Hi, this is Lisa Olson. Hi, now I'm going to get started. And I'm filling in, my name's Lisa Olson. I'm a professor here at Columbia, and I'm filling in for Ed Guo, being a guest host for our Faculty in Focus. And today we are excited to have Santiago Correa, who is one of our brand new professors, who's actually not even here at Columbia quite yet, but he'll be here in January. So with that, I'm just going to let Professor Correa take it over. Thanks, Santiago.
Thanks, Lisa. Let me share my screen. Can you all see my screen?
Looks good.
Yes.
Yes.
Terrific.
Yep. Looks great.
Well, thanks, Lisa, for that introduction. And good morning, everyone. It's a pleasure to be able to share my research with you all today. And today, what I'll be discussing is the development of multifunctional biomaterials through self-assembling nanotechnologies. And so this concept is really at the forefront of nanomedicine, where we often leverage supramolecular interactions to produce nanoparticles with interesting and useful capabilities for drug delivery. And more recently, there's been exciting developments in using nanoparticles themselves as elements of larger meso to macroscopic supramolecular materials. And so overall, these kinds of technologies have been making a difference in the clinic for some time now. So for example, Doxil, the first FDA-approved nanomedicine, was a pegylated liposomal formulation of a common chemo drug that significantly improved its safety profile. Since then, we've seen drugs or nanomedicines like Vyxeos, which have helped to unlock the potential of combination chemotherapy for certain types of hematological cancers. And most recently, we've all seen the incredible capabilities of solid lipid nanoparticles in the development of mRNA vaccines, which have been essential for combating the COVID pandemic. But while these types of nanomedicines have been making strides in the clinic, over the past 20 years or so, we're still seeing this emerging area of nanoparticle-based assemblies. And so this is, again, an area that's, fairly recent and has some really exciting implications for local drug delivery. And the reason for this is that, like other supramolecular macroscopic materials, nanoparticle-based assemblies have useful mechanical properties due to the dynamic and reversible interactions that govern the self-assembly of these materials. And so from a clinical perspective, one of their major advantages is that we can inject these hydrogels directly into target tissues, which has opened a lot of doors in the field of local drug delivery, in particular with regards to immune engineering and tissue engineering. And so I personally have been working in this area of self-assembled nanotechnology for the past 10 years or so now. In my PhD in the Hammond Lab at MIT, I worked on nanocomposites, namely layer by layer nanomaterials. And so while at MIT, I developed new nanofabrication methods. I conducted a screen of nanoparticle surface chemistries, which identified formulations with useful properties for downstream applications in theranostics, immunotherapy, and gene therapy. And these experiences ultimately led me to pursue my postdoctoral work in the Appel group at Stanford, where I started to look at nanoparticles through a different lens, looking at them now as building blocks for these larger macroscopic materials such as hydrogels. And at Stanford, I applied this approach towards improving localized cancer immunotherapy. And I also started to introduce new functionalities into these material systems by engineering the nanoparticle component. And so today what I'm going to be focusing on is primarily my past research, primarily looking at my evaluation of nanoparticle surface chemistries and how that led to new formulations for theranostics and immunotherapy. Also hit on some highlights from my postdoctoral work, using nanoparticle-based hydrogels for cancer immunotherapy. And then I'll round out our discussion with some of the future directions that I'm planning to take in my lab at Columbia with these kinds of material systems. And so I mentioned before this nanocomposite, which I refer to as a layer-by-layer nanomaterial. These are materials that use a process called the layer-by-layer assembly approach, and you can use this to build composite materials either in 2D or in 3D, depending on the kind of substrate that you're working with. And so this image here sort of schematizes this approach of layer-by-layer assembly. It's essentially the sequential deposition of different polymers onto a substrate. In this case, I'm showing a gold nanoparticle, and we're leveraging electrostatic interactions in this particular cartoon to build up multiple layers of different polymers. And by incorporating these different functional materials into a single nanoparticle, we can develop these platform technologies that can carry out complex tasks downstream that have useful implications for biomedical applications. And so that's sort of layer by layer at a high level. I like to break things down a little bit for folks who maybe are not familiar with this technological approach. And we can kind of take a step back and look at LBL nanoparticles in terms of sort of three main parts: the core substrate, the inner polymer layers, and the outer polymer layers. And so we can walk through these step by step. So the core substrate, and this is essentially what determines if you're working in like that 2D layer by layer space or 3D layer by layer space. When we make layer by layer nanoparticles, we're working with charged nanoparticle templates. And those can be any kind of substrate that can be engineered to carry a charge. Here I'm showing a liposome, and the last cartoon I was showing a gold nanoparticle. Essentially any system, any nanotechnology that can be engineered to have a high surface charge can be used for this kind of approach. And this is important because ultimately the properties of that core material are translated over into your final layer by layer or LBL nanoparticle. The next component that I like to explain in detail is the inner polymer layers. And so this is the part of the layer by layer film that is not in direct contact with the biological milieu that these nanoparticles eventually find themselves in when we introduce them into an animal or into a patient. These can be engineered to carry out different or distinct roles within the final particle system. So some examples of what they can do: they can regulate the release of drug from a core material. They can also be therapeutic in and of themselves. So for example, this is generally where we load nucleic acids into layer by layer nanoparticles for gene delivery applications. And then finally, we have the outermost polymer layer. So this is the final layer of your LBL film, and it is what is in direct contact with that biological milieu. And so that means that this outermost layer governs this nanobio interface. And that this is the part of layer by layer nanoparticles that really caught my interest during my PhD, because overall this determines the surface chemistry of this nanoparticle, and that has a really big role in determining the biodistribution of the final formulation. So for example, this outermost coat can help determine the kind of proteins that absorb onto the nanoparticle, forming a protein corona, which has been shown to play an important role determining where these nanoparticles end up in the body. You can engineer these coatings to have anti-fouling properties so you can resist the formation of this corona. And they also play an important role in interacting with cells throughout the body as well. So we were working on developing these nanoparticles to target cancer cells. And so this outermost coating had to be able to engage with cancer cells to ultimately stick onto them and be taken up through particular uptake pathway. As you might imagine, there's also interactions between off-target cells, in this case, immune cells, that are going to be influenced directly by that outermost coating as well. And so something I'd like to really emphasize at this point is that the layer by layer platform is incredibly versatile, because we're able to incorporate really very diverse types of materials into these nanoparticles using the exact same assembly approach. And so that means essentially any polymer system that carries a strong enough net charge can be incorporated into an LBL nanoparticle using the exact same approach. Whereas if you were to use a more traditional nanotechnology, you would need to develop unique chemistries to attach all of these different diverse polymer systems onto these particles in the first place. And so that means we were able to explore things like polysaccharides, polypeptides, as well as synthetic polymers and nucleic acids. Overall, this gives us a really a huge sort of toolkit to explore these kinds of surface chemistry interactions for targeting tumor cells. And so what I was interested in doing was taking advantage of this versatility to develop a library of different LBL nanoparticles and see how those different coatings would essentially translate into different biodistribution profiles. From this library and screen, I was able to ultimately identify a number of different formulations that had useful properties for different biomedical applications like gene therapy and imaging. And so we take a step back and we consider what this library is. So it's a relatively straightforward approach. So we're working with a carboxy modified latex nanoparticle here. So this is a 100 nanometer particle that carries a really strong negative charge on the surface and is very, very brightly fluorescent, which makes it really ideal for these biodistribution studies. So this is our core substrate, if you remember the anatomy from before. We coated these nanoparticles with a cationic polypeptide, polyallarginine, to produce a cationic nanoparticle that would then be compatible with a number of different polyanions. And the reason that we're restricting our library here to negatively charged surfaces is that there's a wealth of literature to indicate that cationic nanoparticles in general are fairly toxic. And so we wanted to limit our library to materials that we would expect to be likely to be biocompatible. And so this library broke down into two major families of surface chemistries. The first family being carboxylated surfaces. And this includes some polypeptides, some synthetic polymers, and a tumor targeting polysaccharide, here shown as sodium hyaluronate or hyaluronic acid. We also wanted to look at our sulfated surface chemistries. And this allowed us to explore a number of native polysaccharides and a few synthetic polymers, including a folate modified heparin sulfate, here shown as 8A, which would allow this family to also have a tumor targeting member. And so overall, we were looking at 10 different surface chemistries, all using the same LBL approach to generate each one of these nanoparticles. And what we wanted to see was how these nanoparticles would interact specifically with ovarian cancer cells. This is an area of that we've been interested in for some time, especially given the relatively poor survival rates for ovarian cancer over the past 30 to 40 years. And the way we approached this was to carry out an in vitro screen. So we looked at how these nanoparticles would interact with a panel of different human ovarian cancer cell lines, 10 to be specific, and how those interactions differed with healthy non-cancerous cells derived from murine splenocytes. After an incubation with these nanoparticles, we took these cells and analyzed them using flow cytometry to look at how much nanoparticle-associated fluorescence we could see in the different cell types. And so looking at this from a very high level, instead of each individual formulation, I like to first show essentially the combined families. So in blue here, we see all the formulations with carboxylated chemistries, and in red, we have all the formulations with sulfated chemistries. In this dark gray bar, we see how many carboxylated nanoparticles are binding to cancer cells, as opposed to healthy cells in this white bar. And so what we can see in this kind of simplified chart is that carboxylated surfaces are mediating the highest amount of nanoparticle accumulation on cancer cells compared to sulfated systems. And then the differential between cancer cells and healthy cells is also greater when we look at these carboxylated surfaces, 13-fold specificity over 6-fold specificity. And so that was promising indicator that, you know, we're going to see some interesting formulations emerge from the carboxylated family that might be exciting for more downstream development. And so I want to take us down a step further. So if this is a very high level consideration, let's step all the way down into a very granular consideration. And so here I'm showing you the raw flow cytometry data for one of our cell lines, KF3, and this is 24 hours after treatment. Here in black, we have our ovarian cancer cells that have not been treated with anything. And so this is essentially our background fluorescence, autofluorescence from the cells. Next, let's take a look at cells that have been treated with an LBL nanoparticle coated with dextran sulfate. And we see this tail emerge here in the red data. This tail is represents our cells that are bound to some of these nanoparticles. And while we do have some cells that have nanoparticles associated with them, the vast majority of cells are ignoring these nanoparticles. And that's in pretty stark contrast to the gray data. So this is our carboxy modified latex, that core material. So the core without any of the LBL film. We see a very broad distribution, a lot more cells taking up these nanoparticles, but we still see some cells that are not taking up the particle at all. And so both of these distributions are pretty different from what we're wanting to see. So here, for example, is the LBL nanoparticle coated with sodium hyaluronate. So this is that tumor targeting carboxylated formulation that I mentioned before. And we see that the entire distribution shifts over to the right, and we have a nice normal distribution. And so we're seeing essentially all the cells are taking up nanoparticles to some degree. And we're seeing in general that uptake is fairly uniform. And like I said, this is to be expected as this has a well-documented tumor targeting capability. So what we were really excited to see was the behavior of two polypeptides. These are carboxylated polymers consisting of polyl aspartate or PLD or polyl glutamate or PLE. And what we can see is that both of these coatings are mediating an order of magnitude improvement in nanoparticle accumulation in these ovarian cancer cells and are outperforming even this sodium hyaluronate system, despite the fact that there is no known receptor ligand interaction that might be occurring with these polypeptides and the surface of these ovarian cancer cells. So overall, this was very interesting to us, but we needed to confirm whether these polypeptides were actually engaging in a specific interaction with these ovarian cancer cells, or if they were just non-specifically very sticky nanoparticles. And so to look at that, we carried out a saturation binding isotherm. And so this is where we expose cells at 4 degrees C to different concentrations of nanoparticles so that we can actually saturate the surface of the cell, since at that temperature the cell can't actually internalize any nanoparticles. This allows us to essentially obtain data that we can fit and extract useful parameters from, such as the apparent KD between the nanoparticle and these ovarian cancer cells. And so here I'm showing the results for our hyaluronic acid coated nanoparticles, which have that specific binding interaction, right? And what we see is the classic curve that is normally associated with a specific interaction. And when we fit this data to our model, we find that our hyaluronic acid nanoparticles have an apparent KD for ovarian cancer cells upper range of 20 picomolar. When we look at this approach using our polypeptide coated nanoparticles, PLD and PLE, again, we see this hallmark curve indicating specific interactions. And when we are able to fit this data and see single digit picomolar affinities for ovarian cancer. Now, in contrast, if we look at some other nanoparticle systems, including another LBL nanoparticle here shown in red, this one coated with that dextran sulfate, or our unmodified core material, or even just pegylated nanoparticles, we see a linear curve that is the hallmark result if you have non-specific interactions. And so in general, we see, very stark distinction between these specific binders and these non-specific binders. So with this, we were confident in saying that our polypeptide coatings are binding through a specific mechanism to these ovarian cancer cells. And our next step was to evaluate if this in vitro finding would actually translate to benefits in vivo for targeted drug delivery. And so for this, we went to an orthotopic model of ovarian cancer. This is a model in an immunocompromised mouse where we can generate widely disseminated tumor nodules throughout the intraperitoneal cavity. We treated these mice with our nanoparticles, this time labeled with an infrared dye, and assessed biodistribution over the next few days. I won't delve deep into this data, but I do want to just show the results for tumor accumulation at 72 hours after treatment. So these are IBIS images. These tissues are egg-sized tumor nodules from the IP cavity, and blue indicates higher concentrations of nanoparticles. And what we can see is very clear higher levels of nanoparticle accumulating in these tumors for our carboxylated systems. So polyglutamic acid, polyaspartic acid, and hyaluronic acid-coated nanoparticles. And that's in contrast to our dextrans sulfate-coated LVL system, as well as a pegylated control nanoparticle system. And so overall, this was very exciting to us since we were seeing these benefits both in vitro and in vivo. The question that I wanted to ask at this point is, what are these different carboxylated nanoparticles doing after they bind to these cells? And so to answer that question, we went back to in vitro studies, specifically looking at cells using confocal microscopy to see the subcellular fate of these different nanoparticles. And so here, what I'm showing you are these confocal images. In dark blue is the actin cytoskeleton, in cyan is the nuclei. Our lysosomal compartments are shown in red, and our nanoparticle signal is shown in green. And what we can see in ovarian cancer cells treated with dextrans sulfate-coated nanoparticles is that at 24 hours, we see these punctate green spots within the interior of a cell, which is what you would expect to see. Generally, when a nanoparticle binds to the surface of a cell, it is quickly internalized. So when we looked at our hyaluronic acid-coated nanoparticles, we weren't surprised to see the same subcellular distribution. This time, the main difference being that we're seeing far more nanoparticle in these cancer cells, which is consistent with our flow cytometry results and our IBIS biodistribution results. Where we were surprised again was with the subcellular trafficking of our polypeptide-coated nanoparticles. So here I'm showing this poly-L-glutamate system. So recall that this is 24 hours after treatment with nanoparticles. And the main difference we see here is that this poly-L-glutamate-coated nanoparticle is being retained on the surface of these ovarian cancer cells rather than being internalized. This is a pretty surprising result. Since, as I mentioned before, normally when nanoparticles bind to the surface of the cell, they are generally internalized within a few hours. We were expecting that the polyaspartate system would be quite similar, since those two polymers differ only by a single methylene group. But we were again surprised to see that this polyaspartate actually behaved pretty distinctly from polyglutamate. And if anything, is almost a hybrid result between the hyaluronic acid and poly-L-glutamate. And by hybrid result, I mean that we're seeing this retention of nanoparticle on the surface. And you can see that as this like beautiful green nanoparticle signal occurring even on the projections between these two cells. But we're also seeing internalization of the nanoparticle into these lysosomal compartments. And through some additional super resolution microscopy work, we ultimately determined that these polyaspartate particles are being internalized through caveolar mechanisms. And so this study overall revealed some really interesting formulations that we could explore with unique subcellular trafficking properties on top of their ability to home towards these ovarian tumors. And so what we'll talk about next is specifically how we continue to develop those two polypeptide coatings. So these are new coatings that hadn't been reported before, that had these useful properties of binding to tumor cells with high affinity, but exhibiting pretty distinct subcellular trafficking profiles. And so first, let's talk about this polyaspartate coating. So again, this is the system that binds both, that ends up both on the outside and on the inside of cells after prolonged incubation periods. And so this was a really opportune formulation for a collaboration that I became involved with at MIT with the Appel Lab. And the goal of this collaboration was to develop a new nano-theranostic material. So a theranostic is a technology that can provide both a diagnostic and therapeutic function. And the goal of this particular approach was to combine the Appel Lab's biosensing peptides with the Hammond Lab's layer-by-layer platform. And the idea here was to have a system that could simultaneously silence a specific gene while also providing urinary-based tumor detection. And so for those who may not be familiar with the Appel Lab's biosensing work, this schematizes the overall approach here. Essentially what they do is they attach these protease-sensitive peptides onto a nanoparticle substrate. And while these peptides are attached to a nanoparticle substrate, the entire construct is too large to be cleared by the kidneys and thus doesn't accumulate in the urine. However, if this nanoparticle enters into a tumor microenvironment where proteases are overexpressed that can cleave these peptides, the cleaved peptides can now be cleared by the kidneys and accumulate in the urine, where you can assay them to determine if an animal or a patient has a tumor. And so the goal was to combine that protease technology with our gene silencing layer-by-layer nanoparticle system. And so I'll give the overall view on this system using that same kind of anatomy as before. Our core substrate here was an anionic liposomal core material, which we had chosen due to the translational relevance of lipid nanotechnology. And then our inner polymer layers consisted of polyoarginine and small interfering, or siRNA. And so these inner layers essentially introduce the gene delivery components in this nanomaterial. The polyarginine allows the nanoparticle to escape into the cytoplasm, and the siRNA ultimately is what prevents expression of a particular target gene. Now, to protect these therapeutic layers, we add our outermost layer, which again is that polyaspartic acid, or PLD, system, which is tumor targeting, but also exhibits that sort of dual localization to the exterior and the interior of the cell. And that's really, really important for this particular system because the protease cleavage has to happen in the extracellular compartment, and the gene silencing has to happen in the intracellular compartments. And so our goal here was to go from our LBL nanoparticle to this hybrid system that included the Appel Lab's biosensor peptides, and then ultimately have this theranostic outcome. Now, the challenge was in this getting from our pure LBL system to this hybrid system, because these biosensor peptides are not charged. They can't be incorporated into an LBL nanoparticle using the LBL approach. And so what we ultimately ended up doing was turning to click chemistry. So we modified around 10% of the carboxyl groups on our polyaspartic acid into propargyl groups, which would be compatible with azide click chemistry. And so using that approach, we now had a way of functionalizing the surface chemistry of these nanoparticles in a way that was really highly flexible. Essentially, any molecule that we could put an azide on, we could incorporate into this nanoparticle now. And so while we ultimately did that to incorporate our biosensors, we also ended up evaluating how these nanoparticles could be further engineered by introducing a targeting ligand known as IRGD and stabilizing polymers, so PEG specifically. And so this approach ultimately worked, and it produced some really nice nanoparticles with uniform properties. And our very first question was whether or not we had preserved that diagnostic functionality. And so for that, we turn to in vivo studies, looking at a number of different tumor models. And what we do here is we inject tumor-bearing or tumor-free mice with our nanoparticle, and then collect the urine 2 hours later and see how much of that protease we can detect in the urine, or the protease-sensitive peptide, I should say. Our first test was with a flank model of pancreatic cancer, and what we saw was this very clear elevated signal in the urine of tumor-bearing mice. We saw a similar result in a metastatic model of colorectal cancer and in a flank model of ovarian cancer. And so overall, we were pretty pleased and convinced that we had preserved the diagnostic functionality in our hybrid nanoparticles. And what we wanted to see next is if all this tinkering with the surface chemistry of the LBL nanoparticle may have changed the subcellular trafficking profile that we were trying to work with. So as you recall, the original polyaspartic acid system was localized on the interior and on the exterior of cells. And we looked at our propargyl modified system. So this is before doing any further click addition. We see a really similar subcellular localization of these nanoparticles. So this green signal that we see kind of arcing around the cell here in this micrograph are surface localized nanoparticles. And then we see nanoparticles localized into these vesicles as well, indicating that we are still gaining entry into the cell. What was really exciting was that we also evaluated this for our click modified nanoparticles. And we saw that when we introduced the IRGD targeting ligand and the biosensor peptide, we are seeing much higher accumulation of nanoparticle in both compartments. So we were preserving that unique subcellular trafficking profile, but just seeing a lot more nanoparticle in both places. And in particular, I'll draw your attention to the intracellular nanoparticle accumulation. So if you compare these vesicles from left to right, you'll notice that there's the vesicles on the right are much larger, almost as if they're engorged, which we hypothesized would actually lead to improved gene silencing, since this would likely lead to leakier vesicles over time. And so to look at that, we went back to our animal models. So here we're working with that flank ovarian cancer model. And this tumor expresses a reporter protein known as luciferase that we can look at through whole animal imaging. And so what we can see here is a negative control. So this is a mouse that was treated just with a sugar solution. We see the steady increase of that reporter over time, since this tumor is rapidly growing. But when we treat the mice with our LBL nanoparticle, so this is our original nanoparticle system without any click modification, we see a nice silencing after a single administration of this nanoparticle. But the exciting result was that when we looked at our click modified nanoparticle that has this diagnostic functionality included, we see the same overall kinetics of silencing, but we see just improved silencing across the board, which really is consistent with what we were seeing with those super resolution micrographs. And so to summarize this particular study, essentially we saw that this unique property of the polyaspartic acid coating could be leveraged to develop this this Theranostic platform that allowed for both gene silencing and urine-based detection of tumors. Moving forward quickly, I just want to very briefly tell you what we ended up doing with this polyl glutamate coating. So this is the system that was retained on the surface of cells. So ultimately, what I did here is I helped a graduate student in our lab spin this out into a new project for targeted immunostimulation. This was a project in the realm of cancer immunotherapy. And so very quickly, I'll just give a quick primer on cancer immunotherapy for those who may not be familiar. So this is a process through which we use the body's own immune system to recognize, seek out, and destroy tumor cells. And we generally schematize this as a cancer immunity cycle that begins with some sort of immunogenic cancer cell death, which releases cancer antigens into the environment, where they interact with antigen-presenting cells that ultimately present them in downstream tumor-draining lymph nodes to effector cells such as T cells. This gives rise to a killer T cell population that can exit the lymph nodes and home in on tumors to recognize them, destroy them, and keep this cycle going. So the entire purpose of cancer immunotherapy is to keep this cycle in motion, and tumors are constantly finding ways to break this cycle down using different immunosuppressive mechanisms. And one of the most promising ways of keeping the cycle going is to treat patients with immunostimulatory compounds. So here I'm showing a cytokine known as interleukin-12 or IL-12. And this is naturally produced by immune cells in your body to really rev up an immune response. And when we administer this to patients, we can see really strong anti-cancer effects, but ultimately, one of the reasons this hasn't been used clinically is that the side effects are overall too toxic. And in fact, in past clinical trials with IL-12, this actually led to some fatalities, which has really slowed down the incorporation of some of these immunostimulatory drugs into the clinic. And so this work became a collaboration with the Hammond Lab at MIT, where they had developed this nanoparticle carrier for IL-12, where they tether the cytokine to the surface of a liposome. This is really effective for carrying this cytokine, but unfortunately, the cytokine is still accessible to immune cells in this configuration. And so where we stepped in was in the development of a layer by layer coating that would hide that IL-12 from the body until the nanoparticle could get to where it needs to go. And so the reason that we ended up using the polyglutamic acid system for this particular application is that we essentially wanted to paint cancer cells with this immunogenic material, because our true target for IL-12 are these bystander immune cells, not the tumors themselves. And in fact, if we used nanoparticles that were internalized by tumor cells, we saw that those tumors would degrade and destroy IL-12 and ultimately reduce the efficacy of the treatment. Overall, this work really inspired me to pursue a postdoc in looking at specifically what materials-based approaches could bring to the field of immunoengineering. And ultimately, that's what brought me to my work at Stanford, working in the Appel Lab. So here, again, I was now starting to look at nanoparticles as a building block for macroscopic materials. And here I started to really look at how this approach could be leveraged to improve local cancer immunotherapy. So I had mentioned before that immunostimulatory drugs can be really toxic, but despite that, the way that we generally administrate these in the clinic is through systemic infusions. And that leads to a lot of off-target effects, which clinically in these immunotherapy settings are generally referred to as immune-related adverse effects. These can be really, really difficult for patients to deal with and can ultimately lead to patients having to stop treatment. And so what I was interested in seeing was whether these nanoparticle-based materials could be used to improve the way that we stimulate the immune system just around the tumor microenvironment itself and the tumor-draining lymph node, and see if that would lead to improvements in efficacy and improvements in the safety profile for these treatments. And so if we go back to this cancer immunity cycle, we can now overlay these injectable hydrogels into this schematic and see some of the ways that we can use this approach to support the cancer immune cycle. We can load drugs into these hydrogels that can repolarize the tumor microenvironment and make it more immunogenic. We can load drugs in that will have an effect on this downstream tumor draining lymph nodes, which is sort of where the training of the immune system occurs. We can even incorporate chemokines into these hydrogels to recruit specific drugs or even deliver immune cells using these technologies. And so what I'll focus on for the remainder of this talk is how we ultimately use this approach to deliver a highly immunostimulatory antibody known as a CD40 agonist. This basically supercharges those antigen-presenting cells and makes them way more efficient at interacting with T cells. Despite the fact that this antibody has been around for quite a long time, it still hasn't been translated into the clinic. And a big reason for that is toxicity issues, similar to what was holding IL-12 back. But in addition to that, this antibody binds to a surface receptor, CD40, that exists on immune cells that are all over your body. So that creates what's called this antigen sink, which means that within 24 hours, this antibody is actually no longer detectable when you're in circulation. And for an antibody, that's actually really bad. Normally, antibodies have circulating half-lives that can be on the order of a month. So these two issues combined have really limited the efficacy of CD40 agonists as a monotherapy. And so what I wanted to do was see if these nanoparticle-based hydrogels could be used to really unlock the potential of CD40 agonists, especially because right now there's a lot of renewed interest in these drugs for combination immunotherapy approaches. And so here, the technology that we're using is a polymer nanoparticle hydrogel. This consists of a dodecyl modified hydroxypropylmethylcellulose polymer and PEG-PLA nanoparticles. When you mix these two components together, you have spontaneous, non-specific interactions between the nanoparticles and the polymer that ultimately lead to this supramolecular hydrogel network forming. Because this happens spontaneously under mild conditions, we can incorporate sensitive cargo like antibody drugs simply by adding them into solution with the nanoparticles prior to mixing the two components together. And so here are some pictures of us injecting these materials through 21 gauge needles. You can see their shear thinning and self-healing properties for these images. So for example, as we apply higher shear forces during injection, the material behaves as a liquid and extrudes through the needle. But after injection, those interactions between the nanoparticles and the polymers reestablish themselves and rapidly regenerate a solid material that can resist moderate forces like the force of gravity. And so we are interested in seeing if this would be useful for improving the local delivery of CD40. And specifically, we wanted to compare it to the method that most immunologists and clinicians were considering for local drug delivery, which is essentially a local bolus injection. We wanted to do this in a highly quantitative way. So this would be really relevant to our collaborators in the clinical and immunology spaces. And so we ended up doing a PET biodistribution study over the course of 2 weeks using radio-labeled CD40 agonists. Here are just two representative images of biodistribution of this antibody 24 hours after administration. On the left here, we see the local bolus, these warmer colors indicating higher concentrations of antibody. And what we can see is that at 24 hours, we're already seeing a lot of antibody getting to off-target tissues like the liver, the spleen, the heart, and distant lymph nodes. As opposed to our polymer nanoparticle hydrogel or PNP hydrogel, where almost all of our drug is still retained in the hydrogel or the surrounding injection site. When we quantified this data over the course of those 2 weeks, we saw that our hydrogel delivery approach increased drug exposure in only 2 tissues, the injection site and the tumor draining lymph node, which in this study was the ipsilateral inguinal lymph node. Everywhere else, we saw decreased amounts of drug exposure and often significantly decreased amounts, including in off-target organs like the spleen and the liver. We hypothesized that this would lead to some improvements in pharmacodynamics of the drug, namely how toxic the drug is and how efficacious the drug is. And to answer those questions, we decided to look at what was happening with cytokines in different parts of the body. And so cytokines like IL-12 that we talked about earlier are proteins that are produced by immune cells to communicate with one another. And so by looking at what's happening with cytokines, we can get a sense for how stimulated or suppressed the immune system is in different parts of the body. And so we carried out Luminex cytokine panels looking at 48 different cytokines in different parts of the body. And to look at toxicity, we decided to see what was happening with circulating cytokines, since a common side effect of CD40 agonists in the clinic is a condition known as cytokine storm syndrome. To look at efficacy, we were seeing how cytokines were being expressed in the tumor draining lymph node, which, as we covered before, is that critical training ground where anti-cancer immune responses are essentially born. And so let's first talk about that circulating cytokine response. So this is the kind of toxic response that we want to modulate. And overall, what we saw was that hydrogels are able to really attenuate the spike of pro-inflammatory cytokines you see from this therapy. So let me walk you through this data. Here we're looking at just 3 pro-inflammatory cytokines: interferon gamma, TNF alpha, and IL-12. In gray, we have an isotype antibody control. So this is our negative control. And you can think of this as sort of a baseline levels of these cytokines when everything is fine and normal. In black, we have our clinical standard control. So this is a systemic administration of a maximum tolerated dose of CD40 agonist. So we can think of this as sort of our ceiling of what's acceptable toxicity due to cytokine levels in circulation. Next up, we look at 2 different local regional drug delivery approaches, both administering twofold what we're administering in this black data. In red, we have the local bolus, and in blue, we have our hydrogel. And what you can see across the board is that the local bolus immediately starts increasing the pro-inflammatory cytokine levels in circulation, indicating that this treatment is more toxic. And some additional data on weight loss that we have bears that out. In contrast, our hydrogels are able to significantly attenuate these levels compared to the clinical standard approach, which is exciting, especially since we're delivering 2 times what we're delivering in this black data. Now, in contrast, when we look at the tumor draining lymph nodes, we see a different story. So let me remind you that this is looking at things 3 days later, 3 days after administration. And what you might notice is now we're seeing similar results for our negative control and our clinical standard control. And what that really indicates is how transient of an immunostimulatory response you see when you administer these antibodies systemically. That has to do with that antigen sink effect I was talking about earlier. That means that these antibodies are quickly depleted. In contrast, both of our local regional approaches are seeing elevated levels of these pro-inflammatory cytokines, with our hydrogels having a slight edge over the local bolus approach. And this really bears out when we look at the histology from these mice. Here we're looking at negative control, our hydrogel with our immunostimulatory drug, and a local bolus of our immunostimulatory drug. And we can see that between these two, the hydrogel and the local bolus, we're seeing a lot more Teal cells, which are those T cell lymphocytes that we want infiltrating into the tumor. Based on that, we wanted to confirm that we are going to see improvements in efficacy. And for that, we turn to this B16F10 melanoma model, which is famously resistant to immunotherapy. We treated these mice 1 time with our hydrogel or a local bolus of CD40 and just saw how that would affect tumor outgrowth. And we saw that when we administer this drug with a hydrogel, we can slow down tumor growth, and we see a moderate but significant benefit in overall survival, where now around 10% of our mice would generally end up as long-term survivors. I mentioned before that we are also quite interested in how this could be used in combination immunotherapy. And so we also looked at how efficacy would change when we combine this with frontline immune checkpoint blockade, PD-L1. And we were really excited to see that when we combine it, local regional approaches with immune checkpoint, we go up to 40% long-term survivors, indicating a really synergistic outcome. Overall, we saw similar results with the local bolus and hydrogel here in this combination setting. But remember that the hydrogel also provided significant safety benefits that we didn't see with the local bolus administration route. And so overall, we were really enthusiastic about this technology as a drug agnostic way to locally administer potent immunostimulatory drugs. And so at this point, we've covered the development of multifunctional nanoparticles, as well as some highly exciting capabilities of nanoparticle-based supramolecular assemblies. In my future research, I'm interested in expanding the capabilities of these kinds of supramolecular materials through the development of multifunctional nanoparticle building blocks. This approach looks to introduce valuable functionality needed to address current challenges in drug and cellular delivery. And my lab at Columbia will specifically be looking to develop a suite of supramolecular materials based on this concept of a multifunctional nanoparticle building block. This includes liposome-based hydrogels for affinity-governed release of drugs, as well as cell-derived biomaterials for cancer vaccine applications, and nanopatterned hydrogels that can mimic lymphatic tissues for T-cell engineering applications. I don't have time to delve into all of these concepts today, but just briefly, I'd like to elaborate a bit on using liposomes to improve local drug delivery. So I recently described a new liposome-based hydrogel system where you can self-assemble this system through hydrophobic interactions between liposomes and amphiphilic polymers. One of the most exciting things about this platform technology is that we can easily engineer the surface chemistry of liposomes to introduce different interactions with proteins that are encapsulated within the hydrogel. So for example, we can engineer it so that the liposomes have minimal interactions with cargo, allowing for passive size-based release, or we can introduce affinity interactions, either through electrostatics or molecular ligands to fine-tune the release rate of these drugs. And what we've seen so far with model protein drugs like GFP, as well as with some antibodies and cytokines, is that we can dramatically tune the release rate of proteins from these hydrogels by engineering and leveraging the surface chemistry of those liposomal building blocks. In the future, I'm hoping to use that approach to coordinate the delivery of multiple protein drugs for cancer immunotherapy. And so at this point, I hope I've been able to get you all excited about the capabilities that multifunctional nanoparticles can bring to the table, not only in the sense of standalone nanoparticle drug carriers, but also for the development of more multifunctional macroscopic supramolecular assemblies. And before we wrap up, I would just like to thank my prior mentors and collaborators, the Hammond Lab at MIT, the Appel Lab at Stanford, and our collaborators in the Bhatia, Irvin, Gambier, and Yaga labs. And with that, I'm happy to take any questions.
So thank you so much, Professor Correa, trying to get those double R's. And I was a little remiss in my introduction, but I just want to say quickly that Santiago stepped up to be one of our BME faculty in focus professors, and all of these lectures are going to be available on our YouTube channel. And I don't see any questions, but I have a sort of a general question, which is, when people graduate from a lab like this, doing work like this, it seems it's very clinically targeted, and very, and it seems like super exciting and successful research that is really leading to what should be great therapies. So do students go to pharmaceutical companies? Do they start startups? I mean, obviously some of them will become professors and go on to postdocs, but how much of a how many steps are there between this research that just looks really so practical and interesting and exciting to actually getting it into the clinic or getting it into pharmaceutical companies that to spend a lot of resources on it?
Yeah, that's a great question. And I would say that there's been a real sort of second wave of enthusiasm for nanotechnology and industry since the success of the Moderna and Pfizer vaccines. And so I would say that, prior to that, I think folks were primarily shuttling into startups to work on what was essentially being considered a bit more risky and cutting edge work. And now I'm seeing a lot of students actually getting recruited directly into some of these mid-size or larger biotech companies like Moderna and Pfizer to develop these things at sort of like, I would say like much closer to bench side or bedside than what we've been seeing before.
Yeah, really interesting. I mean, it just looks like there's so many different paths for this research. So I think we're there is one question. Let me see. Great. Something from Nadine Shaheen here at Columbia. She says, a great presentation with regard to the first LBL material you described, which was partially taken up by cancer cells. Do you know how immune cells and connective tissue cells in these models, for example, fibroblasts, respond to the nanoparticles?
That's a great question. And what we looked at was essentially that flow cytometry results from the first in vitro screen in splenocytes. And so there we saw that some immune cells did bind some of these nanoparticles in different ways, but we didn't carry out the same kind of confocal and super resolution studies on those cells to determine if they were ending up in different areas of immune cells as opposed to cancer cells. And so that's still an open question, I would say.
So great. I don't see any more questions. So I think we can, and it's 11:55, so good time to wrap up. And thanks again, Santiago. And it was such a beautiful talk.
Thanks, Lisa.
Okay, bye, everybody.